Hello there, and welcome to Whispered Knowledge. I'm so glad you found your way here tonight. Maybe you've had a long day, or perhaps you're simply the kind of person who finds comfort in quiet learning, in discovering the remarkable while drifting towards sleep. Either way, you're exactly where you need to be. Tonight, we're embarking on a gentle journey through the most complex structure in the known universe. Not a distant galaxy or a cosmic phenomenon light years away, but something far closer. Something you carry with you every moment of every day, your mind. The human mind is an extraordinary mystery, a 3-pound universe nestled inside your skull, generating thoughts, feelings, memories, and dreams. It allows you to read these words, to feel emotions, to remember your first day of school, and to imagine futures that don't yet exist. Over the next while, as your breathing slows and your body settles into rest, we'll explore 100 remarkable facts about this magnificent organ. Some may surprise you, others may make you reconsider what you thought you knew, and a few might linger in your thoughts as you drift into sleep. Before we begin, if these calm, curious explorations help you unwind or give you a quiet way to end your day, consider liking and subscribing to the channel. It helps others find their way here too, one sleepy soul at a time. Now, let's begin our journey into the mind. The architecture of thought. The human brain contains approximately 86 billion neurons. That's not a rough estimate from decades past, but a number arrived at through painstaking modern research. Each of these neurons is a living cell, a tiny biological computer capable of receiving, processing, and transmitting information. To put this in perspective, if you tried to count all the neurons in your brain, counting one per second without stopping for food or sleep, it would take you nearly 3,000 years. And that's just the neurons themselves, not the connections between them. Each neuron can form thousands of connections with other neurons. These connections are called synapses, and they're where the real magic happens. The total number of synaptic connections in the human brain is estimated to be around 100 trillion. That's more than the number of stars in our galaxy, all contained within the soft tissue inside your skull. Every thought you think, every memory you form, every skill you learn involves specific patterns of activity across these vast networks. When you learned to tie your shoes as a child, specific neurons formed connections. When you remember the face of someone you love, a cascade of electrical and chemical signals flows through particular pathways. These synapses aren't static structures, but dynamic junctions that constantly change. When a synapse is used frequently, it can become stronger, transmitting signals more efficiently. Conversely, synapses that go unused for long periods can weaken or disappear entirely. This process, known as synaptic plasticity, underlies all learning and memory. At a synapse, the electrical signal in one neuron triggers the release of chemical messengers called neurotransmitters. These molecules drift across a tiny gap, the synaptic cleft, and bind to receptors on the receiving neuron. This chemical to electrical to chemical conversion happens thousands of times per second across billions of synapses. The complexity of neural networks means that even small changes can have large effects. Altering the strength of just a few synapses can shift how you respond to a situation, what you remember, or what you decide. Your brain is constantly rewiring itself based on experience, though most of these changes happen beneath conscious awareness. The teenager who practices guitar for hours is physically reshaping the synaptic connections in their motor cortex. The elderly person learning a new language is forming new pathways in language regions. Every experience leaves its trace in the pattern of connections. Your brain uses about 20% of your body's total energy, despite accounting for only about 2% of your body weight. This is remarkable when you consider that the brain is not a muscle, not constantly in motion like your heart, yet it's an energy-hungry organ, generating somewhere between 12 and 25 watts of electrical power. That's enough to power a small LED light bulb. Much of this energy goes into maintaining the electrical gradients across neuron membranes, the very basis of neural communication. Every thought, every perception, every moment of consciousness requires energy. This is why when you're mentally exhausted after a day of intense concentration, it's not just a feeling. Your brain has actually consumed significant resources. The brain's high energy demand means it requires constant oxygen and glucose delivery. Blood flow to the brain is tightly regulated, increasing to active regions and decreasing to less active ones. This is the basis of functional brain imaging techniques like fMRI, which detect changes in blood oxygenation as a proxy for neural activity. Your brain receives between 15 and 20% of the blood pumped by your heart with each beat, ensuring a continuous supply of oxygen and nutrients. Interestingly, the brain's energy consumption doesn't increase dramatically with increased mental effort. Whether you're solving complex mathematics or daydreaming, your brain uses roughly the same amount of energy overall. What changes is the distribution of that energy across different regions. Focused attention shifts energy to task-relevant networks, while the default mode network that activates during rest becomes less active. Even during sleep, the brain maintains high energy consumption, using this time for essential maintenance and consolidation processes. Dehydration affects brain function more quickly than many realize. Just 2% dehydration can impair attention, memory, and other cognitive functions. The brain is approximately 75% water, and even slight fluid imbalances affect its operation. This is one reason staying hydrated improves mental performance. Similarly, blood sugar levels significantly affect cognitive function, as glucose is the brain's primary fuel source. The brain cannot store energy efficiently, making it dependent on continuous supply from the bloodstream. The brain tissue itself has the consistency of soft tofu or gelatin. If you could hold a brain in your hands, you'd be surprised by how delicate it feels, how vulnerable this organ is despite its extraordinary capabilities. This is why the skull exists, a bony fortress protecting the soft neural tissue within. The brain floats in cerebrospinal fluid, which acts as a shock absorber, cushioning it from impacts. Inside this protective shell, the brain orchestrates everything you are. The brain's delicate texture results from its cellular composition. Unlike muscle or bone, brain tissue contains relatively little structural protein. The bulk of the brain consists of cells and the fluid spaces between them. Neurons have long, thin projections called axons and dendrites that reach out to connect with other neurons. These projections are supported by a delicate scaffolding of proteins and are surrounded by glial cells, which outnumber neurons and provide essential support functions. The softness of brain tissue makes it especially vulnerable to certain types of damage. Rapid acceleration or deceleration, as in car accidents or falls, can cause the brain to move within the skull, stretching and tearing neural connections even without direct impact. This is traumatic brain injury, and it can have lasting effects on cognitive function and personality. The brain can also swell when injured or infected, but unlike other organs, it has nowhere to expand within the rigid skull. This makes brain swelling particularly dangerous. Fascinatingly, the brain itself has no pain receptors. Neurosurgeons can operate on conscious patients, stimulating different brain areas without causing pain. The patient might feel sensations, see colors, or even recall memories, but they won't feel pain from the brain tissue itself. Pain receptors exist in the meninges, the protective membrane surrounding the brain, and in blood vessels. This is why headaches occur, but they're not caused by the brain tissue itself hurting. This lack of pain receptors in brain tissue makes sense evolutionarily. By the time something is directly damaging brain tissue, protective pain responses would be too late to help. Your brain can process information faster than the most advanced supercomputers in certain tasks. When you catch a ball, your brain calculates trajectory, distance, and timing in milliseconds, coordinating dozens of muscles to position your hand precisely where it needs to be. When you recognize a familiar face in a crowd, you're performing a pattern recognition task that still challenges the most sophisticated artificial intelligence systems. The brain doesn't work like a computer, processing information in neat, sequential steps. Instead, it operates through massive parallelism with billions of neurons working simultaneously, creating a system of breathtaking complexity and efficiency. This parallel processing gives the brain remarkable capabilities. While a computer must process information sequentially through its central processor, your brain processes visual information, auditory input, bodily sensations, memories, and emotions all at once. Different brain regions specialize in different aspects of processing, but they work in concert through dense interconnections. When you see a dog running toward you, visual cortex processes the image. Memory systems activate knowledge about dogs. Amygdala assesses potential threat. Motor systems prepare responses, all within a fraction of a second. The brain achieves this remarkable processing through elegant neural coding schemes. Individual neurons can represent information through their firing rate, how many electrical impulses they generate per second. But information is also coded in the precise timing of spikes relative to other neurons, in patterns of synchronization across neural populations, and in which specific neurons are active. This multi-layered coding allows rich information representation using biological components that operate on millisecond timescales. However, the brain trades speed for flexibility. While computer processors operate at gigahertz speeds, individual neurons fire at maximum rates of only a few hundred times per second. The brain compensates through parallelism and through clever algorithms that evolved over millions of years. Many perceptual and cognitive processes use approximations and shortcuts that work well in real-world situations, even if they're not perfect. The brain is optimized not for theoretical accuracy, but for practical success in the complex, noisy environments where our ancestors evolved. The brain generates approximately 12 to 25 watts of electrical power. This might not sound like much, but it's enough to illuminate a small LED light. This electricity isn't generated by a single source, but emerges from the collective activity of billions of neurons firing in patterns. When neurons communicate, they generate tiny electrical impulses called action potentials. These travel down nerve fibers at varying speeds, depending on the type of neuron and whether the nerve fiber is insulated with the substance called myelin. Your thoughts quite literally move at different speeds. Brain information travels at different speeds along different pathways. Some neural signals travel along myelinated nerve fibers at speeds exceeding 250 miles per hour, while others move along unmyelinated fibers at less than 2 miles per hour. This variation exists because different types of information require different speeds of transmission. The sensation of pain, for instance, travels more slowly than the reflex that makes you jerk your hand away from a hot surface. Your body is designed to react before you fully feel the pain, a protective mechanism that has saved countless hands from burns throughout human evolution. The brain continues developing well into your 20s. For years, scientists believed the brain was fully developed by late adolescence, but modern neuroimaging has revealed something different. The prefrontal cortex, the region responsible for planning, decision-making, impulse control, and weighing consequences, doesn't finish maturing until around age 25 or even later. This explains a great deal about teenage and young adult behavior. The emotional centers of the brain develop earlier than the rational control centers, creating a period where young people can feel emotions intensely but may struggle to regulate them effectively. This isn't a character flaw, but a neurobiological reality. Memories mysteries. Memories are not stored in single locations, but distributed across networks of neurons throughout the brain. When you remember your childhood home, different aspects of that memory reside in different regions. The visual details might be processed in the occipital lobe, the emotions associated with it in the amygdala and prefrontal cortex, the spatial layout in the hippocampus. When you recall the memory, these distributed networks reactivate in concert, reconstructing the experience. This is why memories aren't like video recordings, but rather like stories we tell ourselves, subject to change and reinterpretation each time we access them. This distributed nature of memory means that different types of information about the same event are stored separately and must be bound together during recall. The binding problem, how the brain reassembles these distributed features into unified experiences, remains an active area of research. Current theories suggest that synchronized neural firing across different regions may provide the mechanism for binding disparate elements into coherent memories. The distributed storage system makes memory both robust and vulnerable. Robust because damage to one brain area doesn't necessarily destroy all aspects of a memory. Other regions may retain pieces that can support partial recall, but vulnerable because the reconstruction process creates opportunities for errors, modifications, and false additions. Each time you remember something, you're not accessing a static file, but actively rebuilding the memory, and this reconstruction can incorporate new information, suggestions from others, or details from similar experiences. Memory researchers distinguish between different types of memory based partly on the brain systems involved. Declarative memory, the ability to consciously recall facts and events, depends heavily on the hippocampus and surrounding medial temporal lobe structures. Procedural memory, the unconscious memory for skills and habits, relies on the basal ganglia, cerebellum, and motor cortex. Emotional memories involve the amygdala's interaction with other memory systems. Each type of memory has evolved to solve different adaptive problems our ancestors faced. The hippocampus plays a crucial role in forming new, long-term memories. This seahorse-shaped structure, buried deep in the temporal lobe, acts as a kind of memory gateway. When you experience something new, the hippocampus helps encode that experience into a form that can be stored long-term. Damage to the hippocampus can leave someone unable to form new memories while leaving old memories intact, a condition known as anterograde amnesia. Perhaps the most famous case is that of Henry Molaison, known for decades simply as HM, who lost his ability to form new long-term memories after surgery removed portions of his hippocampus. He could remember his childhood, but couldn't remember people he met just minutes earlier. His case taught neuroscientists invaluable lessons about how memory works. HM's case revealed that the hippocampus is necessary for forming new declarative memories, but not for storing them long-term or for all types of memory. Despite his inability to form new explicit memories, HM could still learn new motor skills. If you taught him a new hand-eye coordination task, he would improve with practice across multiple sessions, even though he had no memory of previous practice sessions. This dissociation demonstrated that procedural memory, the memory for skills and habits, uses different brain systems than declarative memory. The hippocampus doesn't work alone, but as part of a larger system, including the entorhinal cortex, perirhinal cortex, and parahippocampal cortex. These structures work together to process different aspects of incoming information. The hippocampus itself contains distinct subregions: the dentate gyrus, CA3, CA1, and subiculum, each contributing specialized processing. Information flows through these regions in a specific sequence, undergoing transformations at each stage. Within the hippocampus, special neurons called place cells fire when an animal is in specific locations in its environment. These cells form a cognitive map of space, allowing navigation and spatial memory. The discovery of place cells, which earned John O'Keefe a Nobel Prize, revealed that the brain contains neurons responsive to abstract cognitive variables like location. Grid cells in the entorhinal cortex, which fire in regular spatial patterns, work together with place cells to create a sophisticated navigation system. These discoveries revealed that the hippocampal system doesn't just store arbitrary memories, but actively creates structured representations of experience. Memories are formed simultaneously in the hippocampus and in the cortex. For years, scientists believed memories were first encoded in the hippocampus and then gradually transferred to the cortex for long-term storage. But research from MIT and other institutions has revealed something more nuanced. When you experience something, memory traces form simultaneously in both the hippocampus and the cortex. The cortical memories remain silent for about 2 weeks, gradually maturing while the hippocampal memories are active. Eventually, the cortical memories become stable enough to support recall without the hippocampus, which could help explain why brain damage to the hippocampus affects recent memories more than distant ones. Your brain consolidates memories during sleep. While you rest, your brain doesn't shut down, but shifts into a different mode of operation. During certain stages of sleep, particularly slow wave sleep and REM sleep, the brain replays experiences from the day. Patterns of neural activity that occurred while you were learning something new are reactivated, strengthening the connections between neurons. This process helps transfer memories from temporary storage to more permanent storage, and it also helps integrate new information with existing knowledge. This is why studying before sleep can be more effective than pulling an all-nighter, and why a good night's sleep after learning something new makes a real difference in how well you remember it. During sleep, the hippocampus and neocortex engage in an intricate dialogue. The hippocampus, which rapidly encoded experiences during waking, replays these patterns during sleep. The neocortex, which learns more slowly but stores information more stably, gradually extracts the statistical regularities and general principles from the hippocampal replay. Over time, through repeated cycles of this consolidation process, memories become less dependent on the hippocampus and more firmly established in neocortical networks. This consolidation isn't simply a transfer, but an active reorganization. The brain doesn't just copy hippocampal memories to the cortex. Instead, it extracts the gist, the general principles, the relationships between this new information and existing knowledge. Episodic memories of specific events can be transformed into semantic knowledge, general facts extracted from particular experiences. The details fade while the meaning remains, a process that happens largely during sleep. Different sleep stages contribute differently to consolidation. Slow wave sleep, with its characteristic large, synchronized brainwaves, appears particularly important for consolidating declarative memories, facts, and events. During these slow oscillations, the hippocampus and cortex synchronize their activity, facilitating information transfer. REM sleep with its rapid eye movements and brain activity resembling wakefulness, seems especially important for emotional memory processing and procedural learning. The architecture of a normal night's sleep, cycling through different stages multiple times, may be optimized to support different types of memory consolidation. Research has even shown that you can enhance specific memories during sleep through targeted reactivation. If people learn to associate items with particular sounds or smells, then presenting those cues during sleep strengthens the associated memories. This technique, called targeted memory reactivation, works because the sleeping brain can still process sensory information, and the cues trigger replay of the associated memories. While you can't learn entirely new material during sleep, you can strengthen what was learned while awake. False memories can be remarkably vivid and detailed. Your memory isn't a perfect recording device, but a reconstruction system. Each time you remember something, you're not playing back a stored file, but actively rebuilding the memory from various neural components. During this reconstruction, details can change, be added, or be left out. Studies have shown that people can develop detailed memories of events that never happened, complete with sensory details and emotional responses. These false memories aren't lies or fabrications in the usual sense. To the person experiencing them, they feel absolutely real, indistinguishable from true memories. This has profound implications for eyewitness testimony and for our understanding of how we construct our personal histories. Emotional memories tend to be stronger and more vivid than neutral ones. The amygdala, a small almond-shaped structure deep in the brain, tags emotionally significant events, strengthening their encoding in memory. This is why you might vividly remember where you were during a major news event or a personal crisis, while forgetting mundane details from ordinary days. This isn't just about negative emotions. Joyful experiences can also be etched deeply into memory. The emotional tagging system evolved because remembering events associated with strong emotions, whether danger or reward, offered survival advantages. The tiger that nearly killed you, or the tree that offered abundant fruit, both deserve strong encoding in memory. You forget approximately 50% of newly learned information within an hour if you don't review it. This phenomenon, first documented by German psychologist Hermann Ebbinghaus in the late 1800s, is known as the forgetting curve. Without reinforcement, memories fade rapidly at first, then more slowly over time. However, each time you review information, you reset this curve, making the memory stronger and more resistant to forgetting. This is why spaced repetition, reviewing material at increasing intervals, is far more effective than cramming. Your brain is designed to let go of information it doesn't deem important, conserving resources for what matters. Working memory can typically hold around 4 items at once, though this varies somewhat between individuals and tasks. For decades, psychologists taught that the magic number was 7, plus or minus 2. But more recent research suggests the actual capacity is closer to 4 chunks of information. Working memory is like a mental workspace where you temporarily hold and manipulate information. When you're doing mental arithmetic or following complex directions, you're using working memory. Its limited capacity is why multitasking is largely a myth. Your brain can't truly focus on multiple complex tasks simultaneously. Instead, it rapidly switches between tasks, and each switch costs time and cognitive resources. The feeling of being overwhelmed when juggling too many things simultaneously isn't a personal failing, but a fundamental constraint of working memory. Different types of memory are stored in different brain systems. Episodic memory, the ability to recall specific events from your life, depends heavily on the hippocampus. Semantic memory, your knowledge of facts and concepts, relies more on areas of the temporal cortex. Procedural memory, the kind that lets you ride a bike or touch type, is stored in the basal ganglia and cerebellum and doesn't require conscious recall. This is why someone with severe amnesia might not remember learning to play piano, but can still play beautifully when seated at the instrument. The skills persist, even when the episodic memories of learning them are gone. Your brain maintains multiple memory systems, each optimized for different types of information. The sleeping mind. Sleep is not a uniform state, but consists of distinct stages that cycle throughout the night. When you fall asleep, you don't simply turn off but transition through different states, each with its own brainwave patterns, physiological changes, and functions. Non-REM sleep has 3 stages, from light sleep to deep, slow-wave sleep. REM sleep, where most dreaming occurs, creates a brain state remarkably similar to wakefulness in some ways, yet you're deeply asleep, your muscles temporarily paralyzed, except for your eyes and diaphragm. You dream far more than you realize. While vivid dreams are most associated with REM sleep, dreaming can occur in all stages of sleep. Studies where people are awakened during different sleep stages reveal that mental experiences occur throughout the night. We simply don't remember most of them. Upon waking, the dream you remember might represent only a small fraction of your night's mental wanderings. The brain continues generating experiences even in the absence of external input, creating entire worlds from memory fragments, emotions, and neural noise. REM sleep got its name from the rapid eye movements that occur during this stage. Your eyes dart back and forth beneath closed lids, as if scanning an invisible scene. In many ways, REM sleep creates a paradox. Your brain shows electrical activity similar to wakefulness, yet you're deeply asleep. Your muscles are temporarily paralyzed, a protective mechanism that prevents you from acting out your dreams. This paralysis is controlled by neurons in the brainstem. On rare occasions, this system fails, leading to REM behavior disorder, where people physically act out their dreams, sometimes with dangerous consequences. Most adults need 7 to 9 hours of sleep per night, though individual needs vary. Despite our modern tendency to treat sleep as negotiable, sleep deprivation has serious consequences. After just one night of poor sleep, cognitive functions begin to decline. Attention wavers, reaction times slow, memory formation weakens, and emotional regulation becomes more difficult. Chronic sleep deprivation is linked to increased risk of cardiovascular disease, diabetes, obesity, and neurodegenerative disorders. Your brain uses sleep to clear out metabolic waste products that accumulate during waking hours. One of these is beta amyloid, a protein that forms plaques in Alzheimer's disease. Without adequate sleep, this cleaning process is impaired. During deep sleep, your brain waves slow dramatically. These slow oscillations, occurring less than once per second, sweep across the cortex in coordinated waves. During this stage, the brain is less responsive to external stimuli. It's harder to wake someone from deep sleep than from lighter stages, but far from being inactive, the brain is busy during deep sleep. This is when the body repairs tissues, builds bone and muscle, and strengthens the immune system. The brain consolidates memories and processes emotional experiences. Deep sleep is especially important for children and adolescents, supporting growth and brain development. Your brain can learn during sleep, though in limited ways. Studies have shown that people exposed to sounds or smells paired with learning tasks during waking hours can have those memories strengthened if the same sounds or smells are presented during sleep. This process, called targeted memory reactivation, works because the sleeping brain continues to process information and can replay specific memories when cued. However, you can't learn entirely new complex information during sleep. The brain's learning during sleep builds on what was encoded while awake, rather than creating something from scratch. Sleep deprivation impairs judgment about how impaired you actually are. When you're sleep deprived, you may feel like you're functioning reasonably well, but objective measures tell a different story. Your performance on tasks requiring attention, decision-making, and memory declines significantly. Yet, your subjective sense of impairment doesn't match the reality. This is particularly dangerous in situations like driving, where sleep-deprived drivers may believe they're alert enough to drive safely when they're actually at significant risk of accidents. Microsleeps, brief episodes of sleep lasting just seconds, can occur involuntarily when you're sleep deprived, even when you're trying to stay awake. Lucid dreaming occurs when you become aware you're dreaming while still asleep. In this state, some people can exert varying degrees of control over their dreams, changing the narrative or exploring the dream world intentionally. Brain imaging studies of lucid dreamers reveal increased activity in the prefrontal cortex during lucid dreams compared to regular dreams, particularly in areas associated with metacognition and self-awareness. This frontal activation during REM sleep is unusual, suggesting lucid dreaming creates a hybrid state that shares features of both dreaming and waking consciousness. Some researchers are investigating whether lucid dreaming techniques might be useful therapeutically, perhaps for people with recurring nightmares, though more research is needed. The transition between waking and sleeping hosts unique mental states. Hypnagogia, the transition into sleep, and hypnopompia, the transition out of sleep, can produce vivid imagery, sounds, and sensations. As you drift towards sleep, the thalamus, which normally relays sensory information to the cortex, begins to quiet down. The cortex starts shutting down from front to back, with planning and decision-making areas going offline before sensory processing areas. During this transition, you might experience dream-like thoughts while still partially aware of your surroundings, creative insights emerging from the boundary between conscious control and dream-like free association. Emotions and the deeper brain. The amygdala processes emotions, particularly fear and anxiety, with remarkable speed. This almond-shaped structure can evaluate potential threats and trigger responses before the conscious mind fully registers what's happening. When you jump at a sudden sound or feel a spike of fear at something lurking in shadows, your amygdala has already assessed the situation and initiated a response. This rapid processing evolved to keep us safe. In ancestral environments, the split second gained by not waiting for conscious deliberation could mean the difference between escaping a predator and becoming its meal. The amygdala receives input from all sensory modalities, allowing it to monitor the environment for emotionally significant stimuli. It has two main processing pathways. The fast, direct pathway from the thalamus to the amygdala allows for rapid emotional responses based on crude sensory information. This is the pathway that makes you jump at a garden hose that looks like a snake. The slower pathway routes through sensory cortex, providing more detailed analysis. This is what allows you to realize, a moment later, that it's actually just a hose. The fast pathway prioritizes speed over accuracy, a sensible trade-off when the cost of missing a threat exceeds the cost of a false alarm. Beyond fear, the amygdala processes positive emotions and plays a crucial role in social behavior. It responds to facial expressions, helping you rapidly assess others' emotional states. Damage to the amygdala can impair the recognition of fear in others' faces, while leaving other expressions relatively intact. The amygdala also contributes to reward processing, responding to stimuli associated with positive outcomes. Its role in emotion is broader than just fear, though fear has been most extensively studied because of its clear adaptive significance and the relative ease of studying fear in laboratory animals. The amygdala doesn't just trigger emotional responses, but modulates memory formation. When something emotionally arousing happens, the amygdala enhances the encoding and consolidation of that experience in other brain regions. This is accomplished partly through the release of stress hormones like cortisol and adrenaline, which affect hippocampal processing and signal to the brain that this experience is worth remembering. The emotional tagging of memories explains why you remember emotionally significant events more vividly than mundane ones. Evolution shaped the brain to prioritize memories of situations involving strong emotions, whether danger or opportunity, because these situations had the greatest impact on survival and reproduction. Emotional responses involve both bottom-up and top-down processing. The amygdala sends information upward to the prefrontal cortex, providing emotional content to conscious experience. But the prefrontal cortex also sends signals downward to the amygdala, modulating emotional responses based on context and learned information. This bidirectional communication allows for emotional regulation. When you feel anxiety rising, but use breathing techniques or rational thought to calm yourself, you're engaging prefrontal regions that can dampen amygdala activity. Emotion and cognition aren't separate systems, but deeply intertwined networks constantly influencing each other. The prefrontal cortex helps regulate emotional responses. People with damage to certain areas of the prefrontal cortex may have difficulty controlling emotional impulses, even though they can intellectually understand appropriate behavior. This region, particularly the ventromedial and dorsolateral portions, implements strategies for emotion regulation. When you reappraise a situation to change how you feel about it, or when you suppress an emotional expression, prefrontal areas are actively working to modulate activity in emotion-generating regions like the amygdala. This regulatory capacity develops gradually through childhood and adolescence as the prefrontal cortex matures. Chronic stress physically changes the brain. Prolonged exposure to stress hormones, particularly cortisol, can shrink the hippocampus, impair prefrontal cortex function, and enlarge the amygdala. These changes can create a vicious cycle where stress makes the brain more reactive to stress and less able to regulate emotional responses. The good news is that these changes aren't necessarily permanent. With stress reduction, therapy, and time, the brain can recover much of its normal structure and function. Practices like meditation, exercise, and social connection can help protect the brain from stress-related damage and support recovery. Emotions influence memory formation. Events accompanied by strong emotions are generally remembered better than neutral events. This makes evolutionary sense, as remembering situations that evoked fear or joy helped our ancestors avoid dangers and seek rewards. The amygdala communicates with the hippocampus and other memory systems during emotional events, modulating how strongly memories are encoded. This is why you might remember exactly where you were and what you were doing during a personal crisis or celebration, while countless ordinary moments fade away unremembered. The brain's reward system uses the neurotransmitter dopamine to signal value and motivation. When you experience something pleasurable or anticipate a reward, neurons in areas like the nucleus accumbens and ventral tegmental area release dopamine. This creates feelings of pleasure and motivation, driving you to repeat behaviors that led to rewards. This system evolved to encourage behaviors essential for survival, like eating and reproducing. However, drugs of abuse hijack this system, creating artificial surges of dopamine that can lead to addiction. Understanding this circuitry has been crucial for understanding addiction and developing treatments. Emotions can be contagious. When you see someone smile or cry, Specialized neurons called mirror neurons activate, as if you were performing those actions yourself. This neural mirroring creates a bridge between minds, allowing you to resonate with others' emotional states. This capacity for emotional contagion forms the foundation of empathy. It's why watching someone in pain can trigger discomfort in you, or why laughter spreads through a crowd. Your brain is fundamentally social, shaped by evolution to understand and connect with other minds. The insula processes interoceptive awareness, the sense of your body's internal state. This region receives information about your heart rate, breathing, hunger, and other bodily sensations. It helps generate the bodily feelings associated with emotions. When you feel a flutter of anxiety in your stomach, or warmth spreading through your chest, when with someone you love, the insula is involved in creating these embodied emotional experiences. Damage to the insula can impair emotional awareness, making it harder to identify what you're feeling. This region sits at the intersection of body and mind, translating physical sensations into emotional experiences. Language and communication. Language processing involves distributed networks across the brain, not just discrete language centers. While Broca's area in the frontal lobe and Wernicke's area in the temporal lobe play important roles, language emerges from the coordinated activity of many regions spanning frontal, temporal, and parietal lobes, along with subcortical structures. Different aspects of language, from recognizing speech sounds, to understanding meaning, to producing articulated speech, engage different but interconnected networks. This distributed nature means that language is remarkably resilient. The brain can often compensate for damage to one area by recruiting other regions. Modern brain imaging reveals that the traditional model of language, with Broca's area handling production and Wernicke's area handling comprehension, is oversimplified. Both regions activate during both production and comprehension, though they show some specialization. Moreover, numerous other regions contribute to language processing. The angular gyrus in the parietal lobe plays a role in reading and accessing word meanings. The superior temporal sulcus processes the auditory features of speech. The motor cortex and cerebellum contribute to speech articulation. Language emerges not from the action of individual centers, but from the coordinated activity of distributed networks. These networks communicate through white matter tracts, bundles of myelinated axons connecting distant brain regions. The arcuate fasciculus connects posterior temporal regions with frontal regions. The superior longitudinal fasciculus provides broader frontoparietal connectivity. Recent tractography studies, which use brain imaging to visualize these white matter pathways, have revealed a more complex picture of language connectivity than the simple pathway between Broca's and Wernicke's areas depicted in classical models. Language processing also requires integration with memory, attention, and motor systems. Understanding a sentence requires holding words in working memory while parsing grammatical structure. Naming an object requires retrieving its name from semantic memory. Speaking requires coordinating respiratory and articulatory muscles with millisecond precision. Language isn't a modular system operating in isolation, but an integrative process drawing on cognitive resources from across the brain. Broca's area, traditionally associated with speech production, is more complex than once thought. Located in the posterior inferior frontal gyrus, typically in the left hemisphere, this region is involved in speech articulation and grammatical processing, but it also activates during non-linguistic tasks requiring sequencing and complex motor control. Brain imaging studies reveal that Broca's area contains subregions, some specific for language and others engaged in broader cognitive functions. Damage to Broca's area classically produces non-fluent aphasia, where speech is labored and grammatically simple, though comprehension remains relatively intact. Wernicke's area, associated with language comprehension, lies in the posterior superior temporal gyrus, but defining its exact boundaries has proven difficult. Different researchers identify different regions as Wernicke's area, and modern imaging studies suggest it's better understood as part of a larger network supporting language comprehension rather than as a single discrete center. Damage to this region can produce fluent but meaningless speech. People with Wernicke's aphasia speak smoothly and with normal rhythm, but their words lack coherent meaning. They may also have difficulty understanding spoken and written language, though they're often unaware of their deficits. The arcuate fasciculus, a bundle of nerve fibers, connects frontal and temporal language regions. This white matter tract allows information to flow between areas involved in language comprehension and production. Damage to this pathway can produce conduction aphasia, where people can understand language and speak fluently, but have great difficulty repeating words or phrases. This demonstrates that language function depends not just on the activity of specific regions, but on the connections between them. Recent brain mapping studies have revealed the arcuate fasciculus is actually more complex than a simple cable connecting two areas, with multiple branches supporting different aspects of language processing. Most people have left hemisphere dominance for language. Approximately 95% of right-handed people and about 70% of left-handed people have language functions primarily in the left hemisphere. This lateralization emerges early in development. Even newborns show left hemisphere specialization for processing speech sounds. However, language isn't exclusively left lateralized. The right hemisphere contributes to understanding prosody, the emotional tone and rhythm of speech. It also processes non-literal language like metaphors and jokes. True language comprehension requires both hemispheres working together. Children learn language with remarkable ease compared to adults. There appears to be a critical period for language acquisition, particularly for grammar and accent-free pronunciation. Children exposed to a language before puberty typically master it completely, while those learning later often retain an accent and may struggle with subtle grammatical nuances. This doesn't mean adults can't learn languages, but the process differs neurologically. The brain's plasticity for language learning is highest early in life, gradually declining, though never disappearing entirely. The brain processes written language differently from spoken language. Reading is a recent invention in human history, emerging only a few thousand years ago. The brain doesn't have regions that evolved specifically for reading. Instead, reading repurposes neural circuits that evolved for other functions, particularly visual object recognition. When you read, areas of the left temporal cortex that once specialized in recognizing objects become specialized for recognizing written words. This region, sometimes called the visual word form area, can recognize whole words almost instantly in skilled readers. Learning to read literally changes the brain's organization. Bilingual brains show structural differences from monolingual brains. People who speak 2 or more languages often show increased gray matter density in areas involved in language processing and cognitive control. Bilingualism appears to provide cognitive benefits, particularly in executive functions like attention switching and inhibitory control. Bilinguals must constantly manage 2 language systems, suppressing one while using the other. This mental exercise may strengthen neural networks in ways that provide broader cognitive advantages. Some research has suggested bilingualism might delay the onset of dementia symptoms, though this finding remains an area of active investigation with mixed results across different studies. Perception and reality. Visual processing begins in the retina, but perception emerges in the brain. Your eyes capture light and convert it to neural signals, but these raw signals don't constitute vision. The signals travel through the optic nerve to the thalamus and then to the primary visual cortex in the occipital lobe. From there, information flows along two main pathways: the ventral stream, processing what objects are, and the dorsal stream, processing where objects are and how to interact with them. At each stage, the brain extracts increasingly complex features, from edges and colors to shapes and objects to scenes and meanings. In the retina, photoreceptors convert light into electrical signals. These signals are processed by several layers of neurons before leaving the eye through the optic nerve. Even at this early stage, the visual system begins extracting features like edges, motion direction, and contrast. The retina doesn't passively transmit raw visual data, but performs sophisticated pre-processing, effectively compressing visual information for transmission to the brain. The primary visual cortex, located at the back of the brain in the occipital lobe, contains neurons specialized for detecting simple features like oriented edges and bars. Neurons in this area are organized in columns, with nearby neurons responding to similar orientations, but at slightly different positions in the visual field. This organization creates a retinotopic map, where adjacent positions in the visual world correspond to adjacent neurons in the cortex. Understanding this organization earned David Hubel and Torsten Weisel a Nobel Prize. From primary visual cortex, information flows forward into higher visual areas, each specialized for different aspects of vision. Area V4 processes color and intermediate shape features. Area V5, or MT, specializes in motion perception. Damage to this area can leave someone unable to perceive motion, seeing the world as a series of static snapshots. The ventral stream, sometimes called the what pathway, culminates in the inferior temporal cortex, where neurons respond selectively to complex objects like faces. The dorsal stream, the where and how pathway, extends into parietal cortex, supporting spatial awareness and visually guided action. This hierarchical processing transforms simple features into rich perceptual experiences. But perception isn't purely bottom-up, driven by incoming sensory data. Top-down influences from higher brain areas continuously shape processing at lower levels. Your expectations, attention, and prior knowledge influence what you see. This is why you can find a friend in a crowd more quickly if you know what they're wearing, why ambiguous images can be perceived in multiple ways, and why perception is an active process of interpretation rather than passive reception of information. You don't see the world as it is, but as your brain constructs it. Vision feels immediate and direct, but it's actually an interpretation. Your brain receives incomplete, ambiguous information from your eyes and fills in the gaps based on past experience and expectations. This is why optical illusions work. They exploit assumptions your brain makes about the visual world. The blind spot where the optic nerve exits the retina should create a gap in your vision, but you don't notice it because your brain seamlessly fills it in. Much of what you see is actually prediction and reconstruction. Attention acts as a filter, selecting what enters awareness. At any moment, vastly more sensory information impinges on your receptors than you can consciously process. Attention mechanisms determine which information receives deeper processing and which is filtered out. This is why you can focus on a conversation in a noisy room, tuning out other voices. It's also why you can look directly at something without seeing it if your attention is elsewhere. Inattentional blindness, The failure to notice unexpected objects when attention is engaged elsewhere demonstrates that seeing requires more than functioning eyes. It requires attention. The brain processes different features of a visual scene in parallel. When you see a red ball moving across your field of view, separate populations of neurons process its color, shape, and motion. These features are analyzed in different areas of the visual cortex. The brain then binds these separate features together into a unified perception of a single object. This binding problem, how the brain integrates separately processed features, remains an active area of research. Sometimes binding fails, leading to illusory conjunctions where features from different objects are incorrectly combined. Synesthesia occurs when senses blend in unusual ways. Some people experience sensory cross-activation, where stimulating one sense triggers experiences in another. They might see colors when hearing music or taste shapes. This isn't imagination, but actual perceptual experience, often present from early childhood. Brain imaging studies reveal that synesthetes show heightened connectivity between brain regions that normally process different senses. Synesthesia offers a window into how the brain organizes sensory information and reminds us that perception varies between individuals in fundamental ways. Time perception is subjective and malleable. The brain doesn't have a single clock measuring time. Instead, the feeling of time passing emerges from various neural processes. Highly emotional or novel experiences often feel like they last longer than routine events, and upon reflection, they occupy more space in memory. As you age, years seem to pass more quickly, possibly because fewer experiences are novel and attention demanding. Time perception can also be distorted in the moment. In moments of danger, when attention intensifies and information processing accelerates, seconds can feel stretched out. Your senses don't operate independently, but influence each other. What you see can affect what you hear, and vice versa. The McGurk effect demonstrates this. When you watch someone mouth one syllable while hearing a different syllable, you often perceive a third syllable, a compromise your brain creates. Multisensory integration occurs throughout the brain, with many areas receiving input from multiple senses. This integration usually helps you perceive the world more accurately, but it also means your perception of any single sense is influenced by others. The brain can adapt to sensory deprivation or unusual sensory input. People who lose their sight often show enhanced auditory processing, with visual cortex being recruited to help process sounds. This cross-modal plasticity demonstrates that brain regions aren't as specialized as once thought. Given changed input, they can learn new functions. Similarly, experiments where people wear inverting goggles that flip vision upside down show that within days, the brain adapts and people can function normally. Remove the goggles, and there's another period of adaptation as the brain readjusts to normal input. The learning brain. Neuroplasticity allows the brain to reorganize itself throughout life. For many years, scientists believed the adult brain was relatively fixed, unable to form new neurons or make substantial structural changes. This view has been completely overturned. We now know that the brain retains considerable plasticity across the lifespan. New synaptic connections form. When you learn, existing connections strengthen or weaken based on use, and in some regions, entirely new neurons are born even in adulthood. This plasticity is the foundation of learning, recovery from brain injury, and adaptation to changed circumstances. Multiple mechanisms contribute to plasticity at different scales. At the synaptic level, long-term potentiation and long-term depression strengthen or weaken connections based on patterns of activity. When two connected neurons fire together repeatedly, their connection strengthens. Neurons that fire together wire together, as the saying goes. This Hebbian plasticity forms the cellular basis of associative learning. When you learn that thunder follows lightning, neurons responding to these stimuli form stronger connections, encoding the association. Beyond individual synapses, entire neurons can change their structure. Dendritic spines, the tiny protrusions where neurons receive inputs, can grow, shrink, or disappear based on experience. The complexity of the dendritic tree, which determines how many inputs a neuron can receive, changes with learning and environmental enrichment. Even the white matter, the connections between brain regions, shows plasticity. The degree of myelination increases along pathways that are heavily used, speeding up signal transmission. Perhaps most remarkably, neurogenesis, the birth of new neurons, appears to continue in certain brain regions throughout life. The hippocampus generates new neurons, and these new cells integrate into existing circuits. Physical exercise, learning, and enriched environments seem to promote neurogenesis, while stress and aging may reduce it. The functional significance of adult neurogenesis remains an active area of research, but it represents a striking example of brain plasticity that would have seemed impossible just a few decades ago. Plasticity operates on multiple timescales. Rapid changes in synaptic efficacy can occur within seconds or minutes during learning. Structural changes, like the growth of new dendritic spines, unfold over hours to days. Larger scale reorganization, like the expansion of cortical regions devoted to trained skills, emerges over weeks to months. And some forms of plasticity, particularly during early development, shape brain structure in ways that can last a lifetime. The brain is simultaneously stable enough to maintain your identity and memories, yet plastic enough to learn and adapt continuously. Learning physically changes the brain structure. When London taxi drivers learn the complex layout of city streets, their hippocampi grow larger, particularly in posterior regions involved in spatial memory. When musicians practice their instruments, regions controlling finger movements expand. These aren't just functional changes, but actual structural alterations visible on brain scans. The brain allocates more neural real estate to skills that are practiced. This use-dependent plasticity means that how you spend your time literally shapes your brain. Myelin, the insulation around nerve fibers, continues to change with learning. For decades, myelin was thought to form during development and then remain static. Recent research reveals that myelin is dynamic, increasing along neural pathways that are used frequently. This increased myelination speeds up signal transmission, making well-practiced skills faster and more efficient. The hours of practice that lead to expertise involve not just strengthening synapses, but also optimizing the speed of neural communication through myelination. There's no scientific evidence for distinct learning styles. The popular idea that people are visual, auditory, or kinesthetic learners and should receive instruction matching their style is not supported by research. While people may prefer certain modes of learning, studies consistently show that matching instruction to supposed learning styles doesn't improve outcomes. Effective learning depends more on the nature of the material and the quality of engagement than on matching modality to preference. Understanding this can free educators and learners from unnecessary constraints. Spacing learning over time is more effective than mass practice. Cramming before an exam might help you pass, but the information won't stick long term. Distributed practice, reviewing material at intervals, produces stronger, more durable learning. This spacing effect works because each time you recall information, you strengthen the memory. The slight difficulty of retrieval after a delay actually enhances learning. Your brain needs time to consolidate new information, and spacing allows this consolidation to occur between practice sessions. Testing yourself is more effective than passively reviewing material. The retrieval practice effect shows that actively recalling information strengthens memory more than simply reading it again. This is counterintuitive because retrieval feels harder than review, and learners often mistake ease for learning. But the effort of retrieval is exactly what makes it effective. Each successful retrieval strengthens the memory trace. Frequent low-stakes self-testing is one of the most powerful learning strategies, yet it's underutilized because it feels more difficult than passive review. The brain forms habits through repeated behavior patterns. When you first learn a new skill, it requires conscious attention and effort, engaging prefrontal and parietal regions. With repetition, control gradually shifts to the basal ganglia, structures deep in the brain involved in procedural learning. Once a habit forms, the behavior can occur automatically, freeing up conscious attention for other tasks. This is why you can drive a familiar route while thinking about something else. The automaticity of habits is both a strength and a weakness. Habits free mental resources, but can also be difficult to change because they bypass conscious control. Sleep plays a crucial role in consolidating newly learned skills. Whether you're learning a language, a musical instrument, or a physical skill, practicing before sleep leads to better retention than the same amount of practice followed by waking activity. During sleep, particularly during REM sleep for procedural skills, the brain replays patterns of activity from learning, strengthening the relevant neural pathways. A good night's sleep quite literally helps you master new skills. Consciousness and awareness. Consciousness remains one of the deepest mysteries in neuroscience. We still don't fully understand how subjective experience arises from neural activity. Why does seeing the color red feel like anything at all? Why aren't we just sophisticated information processing systems operating in the dark without any inner experience? This question, sometimes called the hard problem of consciousness, has yet to be satisfactorily answered. We can identify neural correlates of consciousness, brain states that correspond to conscious awareness, but explaining how those neural states generate subjective experience remains elusive. Various theories attempt to bridge this explanatory gap. Global workspace theory proposes that information becomes conscious when it's broadcast widely across the brain, becoming available to multiple cognitive systems. Integrated information theory suggests that consciousness arises from systems that integrate information in specific ways, with the amount of consciousness corresponding to the amount of integrated information the system generates. These theories make testable predictions and have advanced our understanding, but whether they truly explain subjective experience or merely describe its neural correlates remains debated. Studying consciousness scientifically presents unique challenges. Unlike other phenomena, consciousness can only be directly observed from the first-person perspective. You can measure brain activity, behavior, and verbal reports, but these provide only indirect access to conscious experience itself. This creates a fundamental asymmetry. You have direct access to your own consciousness, but can only infer the consciousness of others, including other humans. This doesn't make consciousness scientifically inaccessible, but it requires careful experimental design and interpretation. Certain brain regions and patterns of activity consistently correlate with conscious experience. The thalamocortical system, the interconnected network between thalamus and cortex, seems essential for consciousness. Widespread disruption of this system, as in anesthesia or deep sleep, eliminates consciousness. Within the cortex, posterior regions, particularly parietal and occipital areas, show activity closely linked to conscious content. When you see something, become aware of a thought, or hear a sound, specific patterns of activity in these regions track the content of consciousness. But consciousness isn't localized to a single brain area. It emerges from the coordinated activity of distributed networks. This distributed nature helps explain why consciousness is multifaceted, incorporating sensory experiences, thoughts, emotions, and self-awareness into a unified subjective state. The unity of consciousness, despite being generated by numerous separate neural processes, is itself a puzzle. How does the brain bind disparate processes into a single, unified stream of experience? This binding problem connects to fundamental questions about the nature of the self and subjective experience. You experience consciousness with a delay. Neural processing takes time, and what you experience as the present moment actually reflects information that arrived slightly in the past. Research suggests awareness may lag behind actual events by somewhere between 350 and 500 milliseconds, though estimates vary across different studies and methods. Your brain backfills this delay, creating the illusion of a seamless present. This means that when you consciously decide to perform an action, neural activity associated with that decision began earlier than your awareness of it. This has led to fascinating debates about free will and the nature of conscious decision-making, though neuroscientists interpret these findings in various ways. Split brain studies reveal that the brain can support two separate streams of consciousness. When the corpus callosum, the massive bundle of fibers connecting the hemispheres, is severed to treat severe epilepsy, the two hemispheres lose their main communication pathway. Carefully designed experiments show that each hemisphere can process information independently, and in some situations, they appear to support separate conscious experiences. If you show different images to each hemisphere of a split brain patient, each hemisphere processes its image, and the person reports experiencing only what the left, language-dominant hemisphere saw, though the right hemisphere demonstrates knowledge of its image through nonverbal responses. The default mode network activates when your mind wanders. When you're not focused on external tasks, a specific set of brain regions becomes active, including areas in the medial prefrontal cortex, posterior cingulate cortex, and temporal lobes. This network seems to support self-referential thought, mind wandering, imagining the future, and thinking about others' mental states. Far from being idle, the wandering mind may serve important functions, from consolidating memories to creative problem-solving. The default mode network deactivates during focused external attention and reactivates during rest. Metacognition, the ability to think about your own thinking, involves specialized neural circuits. When you evaluate how confident you are in a memory or decision, or when you monitor your own learning, you're engaging in metacognition. Brain regions, including the prefrontal cortex and precuneus, seem particularly important for these self-reflective processes. Metacognitive abilities vary between individuals and can be improved with practice. Being able to accurately assess your own knowledge and skills is valuable, helping you know when to trust your judgments and when to seek more information. The brain can generate conscious experience without external input. Dreams prove that your brain can create rich, detailed experiences in the absence of sensory information from the world. Hallucinations in various psychiatric and neurological conditions demonstrate the same point. Consciousness doesn't require external input, but can arise purely from internal brain activity. This suggests that what we experience as the external world is actually a kind of controlled hallucination, where sensory input guides and constrains the brain's model of reality but doesn't directly create conscious experience. Anesthesia doesn't simply turn consciousness off like a switch. Different anesthetics work through different mechanisms, affecting various neurotransmitter systems and neural circuits. Under anesthesia, you may cycle through different states, from light sedation, where you remain conscious but drowsy, to deeper states where consciousness fades. General anesthetics seem to disrupt the integrated information processing that characterizes conscious states, fragmenting the normally unified activity of brain networks. Understanding how anesthetics eliminate consciousness might provide clues to understanding consciousness itself. Disorders of consciousness reveal the complexity of awareness. Coma, vegetative state, and minimally conscious state represent different levels of impaired consciousness. Brain imaging sometimes reveals that patients who appear completely unresponsive show brain activity suggesting some level of awareness. In rare cases, patients in vegetative states have demonstrated the ability to answer yes/no questions, using brain activity patterns, thinking of tennis for yes, or imagining walking through their house for no. These findings have profound implications for medical decision-making and our understanding of consciousness. Some people lack an inner voice or the ability to visualize, while many people report an ongoing internal monologue narrating their thoughts. Others report no such experience. Similarly, when asked to visualize an apple, some people see a vivid mental image, while others experience complete aphantasia, the inability to voluntarily create mental imagery. These differences in conscious experience highlight how varied inner mental life can be. Your subjective experience of consciousness may be quite different from others, a reminder that consciousness isn't a single phenomenon, but encompasses diverse mental processes. The social mind. The brain is fundamentally shaped by social interaction. Human brains evolved in social contexts, and much of your neural machinery is dedicated to navigating the social world. From infancy, your brain attunes to social cues. Babies prefer looking at faces over other visual stimuli. They can distinguish different facial expressions within the first months of life. This social orientation isn't learned, but reflects brain systems specialized for social processing. The social brain network includes regions like the superior temporal sulcus, which processes biological motion and helps you recognize actions and intentions from movement patterns. The fusiform face area specializes in face recognition, allowing you to quickly identify individuals and read subtle facial expressions. The temporal poles store social knowledge about people and social situations. These regions work together to support the complex social cognition humans require to navigate group life. Social neuroscience has revealed that the brain processes social information using partially distinct circuits from those used for non-social information. When you think about people, different brain regions activate than when you think about objects or abstract concepts. This special status of social information makes sense from an evolutionary perspective. For our ancestors, understanding social dynamics, predicting others' behavior, and navigating complex social hierarchies could mean the difference between thriving and failing, between finding mates and being excluded from the group. Social exclusion activates brain regions overlap with physical pain networks, suggesting that the brain treats social connection as a fundamental need, comparable in importance to physical safety. Studies where people are excluded from simple ball-tossing games show activation in the anterior cingulate cortex and anterior insula, regions also active during physical pain. This overlap isn't metaphorical, but reflects shared neural mechanisms. For social species like humans, separation from the group could be life-threatening, so evolution wired social pain into the same systems that signal physical danger. Mirror neurons activate both when you perform an action and when you observe someone else performing it. First discovered in macaque monkeys, these neurons appear to provide a direct experiential understanding of others' actions. When you see someone reach for a cup, neurons involved in your own reaching behavior activate. This mirroring seems to extend beyond actions to emotions and sensations. Seeing someone in pain activates some of the same brain regions active when you experience pain yourself. Based on these findings, some researchers propose that mirror systems may form part of the neural foundation of empathy, allowing you to resonate with others' experiences, though the exact role of mirror neurons in human social cognition continues to be investigated. Theory of mind, the ability to attribute mental states to others, involves specialized neural networks. Understanding that others have beliefs, desires, and knowledge different from your own is a crucial social cognitive skill. This ability typically emerges around age 4 in most developing children, though the exact timing can vary. Brain regions including the medial prefrontal cortex and temporoparietal junction show increased activity when people think about others' mental states. Autism spectrum disorders often involve difficulties with theory of mind tasks, and neuroimaging studies show altered activity patterns in these brain regions. The brain processes social rejection using some of the same circuitry as physical pain. Experiences of social exclusion or rejection activate the anterior cingulate cortex and insula, regions also involved in processing physical pain. This overlap isn't just metaphorical. Social pain and physical pain share neural mechanisms, which makes evolutionary sense given that for social species, exclusion from the group could be life-threatening. The brain treats social connection as a fundamental need, comparable to food or safety. Oxytocin, often called the bonding hormone, modulates social behavior. This neurotransmitter is released during childbirth, breastfeeding, and sexual activity, but also during positive social interactions. Oxytocin increases trust, enhances face recognition, and promotes bonding. However, its effects are more complex than simple social warmth. Oxytocin can also enhance in-group favoritism and defensive responses to outgroups. It seems to amplify social salience generally, making social cues more important and emotionally impactful. Your brain represents social hierarchies. Primates, including humans, navigate complex social hierarchies, and the brain contains specialized systems for processing social rank. The prefrontal cortex and striatum show different patterns of activity when thinking about social superiors versus subordinates. People at different social positions show distinct patterns of brain activity and stress hormone levels. The experience of low social status affects the brain's stress response systems, potentially contributing to health disparities across socioeconomic gradients. The brain is more engaged when information is socially relevant. When learning something, you'll remember it better if it's presented in a social context or related to yourself. The medial prefrontal cortex, a region involved in self-referential processing, activates more strongly when you're learning information relevant to you personally. This self-reference effect demonstrates that the brain doesn't treat all information equally, but prioritizes socially and personally relevant information. Loneliness affects brain function and structure. Chronic loneliness, the persistent perception of social isolation, has neurobiological consequences. It activates stress response systems, affects immune function, and alters brain regions involved in social cognition and emotional regulation. Lonely individuals show heightened vigilance for social threats, creating a self-perpetuating cycle where loneliness makes social interactions more difficult. The brain is shaped by social connection, and prolonged social isolation can have effects as serious as physical health problems. The aging mind. The brain grows dramatically in early childhood. At birth, your brain is about a quarter of its adult size. By age 3, it reaches about 80% of adult size, and by age 6, it's about 90%. This growth involves not just getting bigger, but massive reorganization. In early development, the brain actually produces more synapses than it will ultimately keep, then prunes connections that aren't used frequently. This pruning refines neural networks, making them more efficient and specialized. The brain you have as an adult is shaped as much by pruning as by growth. Synaptic density peaks at different ages in different brain regions. In visual cortex, synapse numbers peak around 8 months and then decline. In prefrontal cortex, the peak occurs much later, during early childhood, with pruning continuing through adolescence and into the mid-20s. This protracted development of prefrontal regions, which support planning, impulse control, and decision-making, explains why these abilities mature gradually across childhood and adolescence. The developing brain shows remarkable plasticity, which allows learning but also creates vulnerability. Critical periods exist for certain abilities, windows when the brain is especially sensitive to particular types of experience. For vision, the critical period occurs in early childhood. Children who have cataracts removed after this period may never develop normal vision, even though their eyes function properly. For language, there's a sensitive period extending through childhood into adolescence. Children exposed to language early acquire it effortlessly, while later learners must exert conscious effort and often retain accents and grammatical imperfections. Environmental influences during development can have lasting effects. Childhood malnutrition, exposure to toxins, chronic stress, or lack of cognitive stimulation can alter brain development in ways that persist into adulthood. Conversely, enriched environments with opportunities for learning, exploration, and social interaction promote healthy brain development. These environmental effects interact with genetic factors, with some individuals more resilient and others more susceptible to environmental influences. The developing brain isn't a passive recipient of genetic instructions, but an active system shaped by the continuous interplay of genes and experience. Different cognitive abilities peak at different ages. While it's true that some mental functions show age-related changes, others improve or remain stable. Processing speed and certain types of memory tend to peak in the 20s and then gradually decline. But vocabulary continues growing well into the 60s or beyond. Emotional regulation and the ability to see multiple perspectives on problems often improve with age. Crystallized intelligence, accumulated knowledge, and skills typically doesn't decline and may continue increasing throughout much of life, even as fluid intelligence, the ability to reason with novel information, shows gradual decreases. The aging brain compensates for declines in some areas by recruiting additional regions. Brain imaging studies show that older adults often activate more extensive brain networks than younger adults when performing the same tasks. This might represent compensation, with the older brain recruiting additional resources to maintain performance. In some cases, bilateral activation, using both hemispheres for tasks that younger brains accomplish with one hemisphere, helps older adults perform as well as younger adults. Cognitive reserve refers to the brain's resilience to aging and damage. People with more education, mentally stimulating careers, and active social lives tend to show less cognitive decline with age. They may even tolerate more brain pathology, like Alzheimer's plaques and tangles, before showing symptoms. This doesn't mean they don't develop these changes, but that their brains can better compensate. Building cognitive reserve throughout life through learning, social engagement, and mental challenges may help protect against cognitive decline. Physical exercise benefits the aging brain. Cardiovascular exercise increases blood flow to the brain, promotes the growth of new neurons in the hippocampus, and supports the production of neurotrophic factors that help neurons survive and form connections. Studies show that regular exercise can improve memory and slow cognitive decline. Even starting exercise late in life provides benefits. The brain retains plasticity and responsiveness to experience throughout the lifespan. The brain changes structurally with age. Starting around age 30, the brain begins to shrink slightly, a process that accelerates in older age. Certain regions, particularly the prefrontal cortex and hippocampus, are especially vulnerable to age-related atrophy. White matter, the bundles of myelinated nerve fibers connecting brain regions, also shows age-related changes. However, these structural changes don't affect everyone equally, and brain structure in late life is influenced by genetics, lifestyle, and health factors throughout life. Wisdom may involve unique patterns of brain activity. While difficult to define scientifically, wisdom seems to involve integrating cognitive and emotional information, seeing issues from multiple perspectives, and making judgments that consider long-term consequences. Some research suggests that wisdom involves coordination between brain systems that often work in opposition, such as those supporting analytical thinking and those supporting empathy. Older adults who score high on measures of wisdom show distinct patterns of brain activity and connectivity. The aging brain becomes more vulnerable to certain disorders. Alzheimer's disease, the most common form of dementia, primarily affects older adults. It involves the accumulation of abnormal proteins in the brain, leading to neuron death and progressive cognitive decline. Parkinson's disease, which affects movement through degeneration of dopamine-producing neurons, also increases with age. While aging itself doesn't cause these disorders, age-related changes in cellular repair mechanisms and accumulated damage over decades increase vulnerability. The darker territories. Mental illnesses involve alterations in brain function and often structure. Depression is associated with changes in neurotransmitter systems, particularly serotonin, norepinephrine, and dopamine, as well as structural changes, including hippocampal atrophy. Schizophrenia involves altered dopamine signaling and abnormalities in brain structure and connectivity. Anxiety disorders show hyperactivity in the amygdala and altered prefrontal regulation of emotional responses. These conditions aren't simply psychological, but have clear neurobiological bases. Understanding this has reduced stigma and led to more effective treatments. The neurobiological understanding of mental illness has evolved considerably. Early theories focused on chemical imbalances, particularly neurotransmitter deficiencies. While neurotransmitters remain important, current models recognize mental illness as involving complex interactions between neurotransmitter systems, neural circuits, genetic factors, environmental influences, and developmental trajectories. Depression, for instance, isn't simply a serotonin deficiency. It involves alterations in stress response systems, inflammation, neuroplasticity, and the function of multiple brain networks supporting emotion regulation, reward processing, and cognitive control. Brain imaging has revealed structural and functional differences associated with various mental illnesses. People with major depression often show reduced volume in the hippocampus and prefrontal cortex, regions important for memory and emotion regulation. Schizophrenia is associated with enlarged ventricles and reduced gray matter in several regions. These structural differences reflect underlying cellular changes, including altered neuronal density, reduced dendritic complexity, and glial cell abnormalities. While these findings don't provide simple diagnostic markers, they demonstrate that mental illnesses have physical manifestations in the brain. Treatment approaches increasingly target specific neural mechanisms. Antidepressants modulate neurotransmitter systems, but also promote neuroplasticity and neurogenesis. Psychotherapy, particularly cognitive behavioral therapy, produces measurable changes in brain function, normalizing activity in regions showing abnormal patterns. Newer treatments like transcranial magnetic stimulation directly modulate activity in specific brain regions. Understanding the neurobiology of mental illness opens pathways to more targeted, effective interventions. Addiction fundamentally alters the brain's reward system. Drugs of abuse cause massive surges of dopamine in reward circuits, far exceeding natural rewards. With repeated exposure, the brain adapts, becoming less sensitive to dopamine. This tolerance means the person needs more of the drug to feel its effects. Meanwhile, natural rewards lose their appeal. The prefrontal cortex, involved in impulse control and decision-making, shows altered function in addiction. These changes help explain why addiction is so difficult to overcome through willpower alone. It involves persistent changes to brain circuits underlying motivation and control. The neurobiology of addiction involves multiple brain systems. The ventral tegmental area and nucleus accumbens form the core of the reward circuit. These regions normally reinforce behaviors essential for survival by releasing dopamine in response to food, sex, and social connection. Addictive drugs hijack this system, creating artificial dopamine surges that dwarf natural rewards. Over time, repeated drug use causes the reward system to become less responsive, a state called downregulation. The individual needs the drug just to feel normal, as natural rewards no longer sufficiently activate the blunted reward system. Beyond the reward system, addiction involves the prefrontal cortex's role in executive function and self-control. Imaging studies show reduced activity in prefrontal regions in addicted individuals, particularly during tasks requiring inhibitory control. This reduced prefrontal function makes it harder to resist cravings, even when the person intellectually understands the negative consequences of continued use. The amygdala, involved in stress and emotional responses, becomes hyperactive, contributing to the negative emotional states during withdrawal that drive continued drug use. Importantly, these brain changes can persist long after drug use stops. The neuroadaptations underlying addiction are remarkably stable, which explains why relapse remains a risk, even years into recovery. However, the brain retains plasticity, and with time and appropriate treatment, recovery is possible. Understanding addiction as a brain disorder has shifted treatment approaches from moral judgments toward medical interventions, addressing the underlying neurobiology. Chronic stress can shrink the hippocampus and enlarge the amygdala. Prolonged exposure to stress hormones has toxic effects on hippocampal neurons, and brain imaging studies consistently show smaller hippocampal volume in people with chronic stress or depression. Meanwhile, the amygdala, central to fear and anxiety responses, becomes overactive and can increase in size. These changes create a brain more prone to anxiety and less able to regulate stress responses, potentially contributing to mental health disorders. Stress reduction and treatment can reverse some of these changes. Post-traumatic stress disorder involves persistent alterations in fear circuits. After trauma, most people experience heightened stress responses that gradually normalize. In PTSD, These responses persist, with the amygdala remaining hyperactive and the prefrontal cortex showing reduced ability to regulate fear responses. The hippocampus, involved in contextualizing memories, is often smaller in PTSD. These changes mean that trauma memories remain emotionally vivid and can be triggered by reminders, producing intense fear responses, even in safe situations. Understanding the neurobiology of PTSD has led to more effective treatments targeting these specific circuits. Cognitive biases represent systematic errors in thinking. Your brain uses mental shortcuts, called heuristics, to make decisions efficiently. Usually these work well, but they can lead to predictable errors. Confirmation bias leads you to notice information supporting your beliefs while ignoring contradictory evidence. The availability heuristic causes you to overestimate the likelihood of events you can easily recall. These biases arise from how the brain processes information and makes predictions. They're not signs of irrationality, but features of a system optimized for speed and efficiency rather than perfect accuracy. Hundreds of cognitive biases have been documented, and they affect every aspect of thinking and decision-making. The anchoring effect causes you to rely too heavily on the first piece of information you receive, even if it's irrelevant. The sunk cost fallacy leads you to continue investing in failed projects because of what you've already invested. Hindsight bias makes past events seem more predictable than they actually were. The "I knew it all along" phenomenon. Understanding these biases doesn't make you immune to them, but it can help you recognize when you might be making systematic errors. Many cognitive biases reflect reasonable heuristics that work well in most situations, but fail in specific contexts. The representativeness heuristic, judging probability based on how representative something seems of a category, usually serves you well, but it can lead to errors like the conjunction fallacy, believing specific scenarios are more likely than general ones when the opposite is logically true. These biases reveal the brain's preference for fast, intuitive judgment over slow, logical analysis. Most of the time, speed trumps perfect accuracy in real-world decision-making. The placebo effect demonstrates the brain's power over the body. When people believe they're receiving treatment, they often improve, even if the treatment is inert. Brain imaging reveals that placebos can activate the same neural pathways as actual medications. Placebo analgesia, pain relief from fake painkillers, involves real activation of the brain's pain-modulating systems, including the release of endogenous opioids. The placebo effect isn't imaginary, but reflects genuine changes in brain function driven by expectation and belief. The mechanisms underlying placebo effects are complex and involve multiple brain systems. Expectation and conditioning both contribute. If you've previously experienced pain relief from a pill, taking a similar-looking pill can trigger the same brain responses, even if the new pill contains no active ingredients. The prefrontal cortex, involved in expectation and prediction, communicates with brainstem regions that modulate pain signals. This top-down modulation can genuinely reduce pain perception, not just change reports of pain. Interestingly, placebos work even when people know they're taking placebos in some cases. Open-label placebo studies, where participants are told they're receiving inactive pills, still show therapeutic benefits for some conditions. This suggests the placebo effect isn't purely about deception, but involves ritual, hope, and the therapeutic context. The act of receiving treatment, combined with the expectation that it might help, can trigger genuine biological responses. The placebo effect extends beyond pain. Placebo responses occur in depression, Parkinson's disease, irritable bowel syndrome, and many other conditions. Interestingly, Some analyses have suggested the magnitude of placebo effects may be increasing over time in clinical trials, possibly due to changing cultural beliefs about medical treatment, though this trend continues to be studied. Understanding placebo mechanisms isn't just about controlling for them in clinical trials, but about potentially harnessing the brain's self-healing capabilities to enhance medical treatments. Depression isn't simply sadness, but involves disruptions across multiple brain systems. Beyond emotional symptoms, depression affects motivation, pleasure, energy, sleep, appetite, and cognition. Neuroimaging reveals reduced activity in prefrontal regions and altered function in the amygdala and other limbic structures. Neurotransmitter systems, including serotonin, norepinephrine, and dopamine, show abnormalities. Recent research has also implicated inflammation and disrupted neuroplasticity. Depression is a complex brain disorder affecting multiple systems, which is why treatment often requires targeting several mechanisms simultaneously. The symptoms of depression reflect dysfunction in specific neural circuits. Anhedonia, the inability to experience pleasure, relates to reduced activity in reward circuits involving the nucleus accumbens and ventral tegmental area. These regions show blunted responses to rewards in depressed individuals. The negative thought patterns characteristic of depression involve altered activity in the default mode network, particularly rumination involving excessive self-focused thought. Cognitive symptoms like difficulty concentrating and making decisions reflect reduced prefrontal cortex function. Sleep disturbances in depression, including both insomnia and hypersomnia, involve disrupted circadian rhythms and altered sleep architecture. Depressed individuals often show abnormalities in REM sleep, including earlier onset and increased REM density. These sleep disruptions interact bidirectionally with depression. Disturbed sleep worsens mood, and depression disrupts sleep. Treating sleep problems can sometimes improve depressive symptoms, highlighting the interconnection between different brain systems. The neuroplasticity hypothesis of depression suggests that chronic stress and depression reduce the brain's ability to generate new neurons and form new synaptic connections. Levels of brain-derived neurotrophic factor, a protein supporting neuronal growth and survival, are reduced in depression. Antidepressant medications may work partly by restoring neuroplasticity, promoting neurogenesis in the hippocampus, and supporting the formation of new connections. This process takes time, which may explain why antidepressants typically require several weeks to show benefits. Inflammation has emerged as an important factor in depression. Many depressed individuals show elevated levels of inflammatory markers in their blood and cerebrospinal fluid. Inflammatory cytokines can affect neurotransmitter metabolism, reduce neuroplasticity, and alter neural circuit function. Some forms of depression may be primarily inflammatory in nature, and anti-inflammatory treatments show promise for certain patients. This represents a shift from viewing depression purely as a chemical imbalance toward understanding it as involving complex interactions between neural, hormonal, immune, and metabolic systems. The heterogeneity of depression, the fact that it manifests differently across individuals, likely reflects different underlying neurobiological mechanisms. Some people's depression may primarily involve serotonin dysfunction, others dopamine or norepinephrine, others inflammation or stress hormone dysregulation. This biological heterogeneity helps explain why treatments that work well for some people are ineffective for others. Future approaches may involve better matching treatments to individuals' specific neurobiological profiles, moving toward personalized medicine for mental health. Obsessive-compulsive disorder involves abnormal communication between the cortex and basal ganglia. Brain circuits that normally help you detect potential problems and disengage from unnecessary behaviors become stuck in OCD. The orbitofrontal cortex, anterior cingulate cortex, and caudate nucleus show abnormal activity. These regions form a loop that, when functioning normally, helps you adjust behavior based on feedback. In OCD, this loop becomes overactive, creating persistent doubt and compelling repetitive behaviors to neutralize perceived threats. Understanding this circuitry has led to both medication and psychotherapy approaches targeting these specific brain systems. The cortico-striato-thalamo-cortical circuits in OCD create a self-perpetuating cycle. The orbitofrontal cortex detects potential problems or threats. Normally, the basal ganglia help filter these signals, allowing you to disengage from concerns once you've addressed them. In OCD, this filtering mechanism malfunctions. The caudate nucleus fails to suppress the worry signal, so it keeps cycling back through the thalamus to the cortex. This creates the persistent, intrusive thoughts characteristic of OCD. The compulsive behaviors in OCD represent attempts to reduce the anxiety generated by obsessive thoughts. When someone with OCD washes their hands repeatedly or checks locks multiple times, they're trying to satisfy the persistent doubt signals from their malfunctioning neural circuits. Unfortunately, the relief is temporary. The circuit dysfunction remains, so the doubts and compulsions return. This is why treatment focuses on breaking the cycle rather than simply performing the compulsions. Effective treatments for OCD target these neural circuits through different mechanisms. Selective serotonin reuptake inhibitors, often at higher doses than used for depression, modulate activity in the cortico-striatothalamo-cortical loops. Cognitive behavioral therapy, particularly exposure and response prevention, helps patients resist compulsions, gradually retraining the neural circuits. Brain imaging studies show that successful treatment, whether medication or therapy, normalizes activity in the overactive brain regions. This demonstrates that psychological interventions can produce measurable biological changes, and biological treatments can improve psychological symptoms. The two are inseparable aspects of the same neural dysfunction. Autism spectrum disorders involve differences in brain development and function. Autistic individuals often show differences in how various brain regions communicate, particularly between areas involved in social cognition. Some research suggests reduced connectivity between distant brain regions, but increased local connectivity. The social brain network, including regions like the superior temporal sulcus and medial prefrontal cortex, shows altered activity patterns. Autism isn't a single condition, but a spectrum, reflecting diverse neurobiological differences that affect social communication and behavior in varied ways. The neural differences in autism emerge early in development and affect multiple brain systems. Studies of brain growth patterns show that some autistic children experience accelerated brain growth in the first few years of life, followed by slower growth later. This altered developmental trajectory may contribute to the formation of different neural networks. The connectivity differences, with stronger local connections but weaker long-range connections, may underlie both the challenges and strengths associated with autism. Sensory processing differences are common in autism and reflect altered neural responses to sensory input. Many autistic individuals experience sensory sensitivity, finding certain sounds, textures, or lights overwhelming. Brain imaging reveals that sensory cortices in autistic individuals may respond more intensely to stimulation. The filtering mechanisms that typically help neurotypical people tune out irrelevant sensory information may work differently in autism, leading to sensory overload in busy environments. The special interest and attention to detail characteristic of many autistic individuals may relate to the pattern of neural connectivity. Enhanced local processing with strong connections between nearby neurons could support detailed, focused attention on specific topics or perceptual features. The challenge is that this comes with trade-offs in global integration, making it harder to shift attention flexibly between different aspects of a situation, or to grasp broader social contexts that neurotypical individuals process intuitively. The future brain. Brain-computer interfaces are becoming increasingly sophisticated. Researchers have developed systems that allow paralyzed individuals to control robotic arms or computer cursors using only their thoughts. These work by recording neural activity, usually through electrodes implanted in motor cortex, and decoding the intended movements. As decoding algorithms improve and electrode technology advances, these interfaces become more accurate and capable. Future systems might restore communication and mobility to people with severe paralysis or neurodegenerative diseases. Neurofeedback allows people to modulate their own brain activity. By receiving real-time feedback about neural activity through brain imaging or EEG, people can learn to control specific patterns of brain activity. This has been explored as a treatment for various conditions, from ADHD to chronic pain. While results have been mixed, the principle demonstrates that with the right feedback, people can develop some conscious control over brain processes normally considered automatic. Optogenetics allows researchers to control specific neurons with light. By genetically engineering neurons to express light-sensitive proteins, researchers can activate or silence specific cell populations with millisecond precision. This technology has revolutionized neuroscience research, allowing scientists to test causal relationships between neural activity and behavior. While currently used only in animals, optogenetic principles might eventually inform new treatments that target specific neural circuits. Transcranial magnetic stimulation can modulate brain activity non-invasively. By applying magnetic fields to the scalp, TMS can stimulate or inhibit neural activity in underlying brain regions. It's FDA approved for treating depression and is being investigated for numerous other conditions. The ability to temporarily alter brain function non-invasively offers both research opportunities and therapeutic potential. Understanding precisely what TMS does to neural circuits and how to optimize its effects remains an active area of investigation. We're beginning to decode thoughts from brain activity. Machine learning algorithms can identify what images someone is viewing, what words they're hearing, or even reconstruct basic aspects of their dreams based on brain scanning data. While we're far from reading minds in the science fiction sense, the ability to decode increasingly complex mental content from neural activity is improving rapidly. This raises both exciting possibilities for communication with nonverbal patients and important ethical questions about mental privacy. Neuroscience is revealing the biological basis of individual differences. Why do people differ in personality, intelligence, and susceptibility to mental illness? Brain imaging and genetics are beginning to provide answers. Studies are identifying brain connectivity patterns associated with different personality traits, genetic variants that affect brain structure and function, and how experience interacts with biology to shape individual development. Understanding the neurobiology of individual differences might eventually allow more personalized approaches to education and medicine. We don't yet understand consciousness. Despite decades of neuroscience research, the fundamental question of how subjective experience arises from neural activity remains unsolved. We can identify neural correlates of consciousness, and understand much about brain function, but explaining why neural activity feels like anything at all, why there's something it's like to be you experiencing your life, remains mysterious. This question sits at the intersection of neuroscience, philosophy, and physics, and solving it may require new conceptual frameworks. The brain contains approximately 86 billion neurons and 100 trillion synaptic connections, yet we've mapped only a fraction of this circuitry. Major initiatives like the Human Connectomy Project aim to map brain connectivity, but we're still far from understanding the complete wiring diagram. Even in simpler organisms like the roundworm C. elegans, whose entire nervous system has been mapped, we can't fully predict behavior from neural connections alone. The relationship between structure and function in brains remains incompletely understood. Neuroplasticity offers hope for recovery from brain injury. While the brain doesn't regenerate as completely as some other organs, its capacity for reorganization means that function can often be restored after injury through rehabilitation. The undamaged portions of the brain can sometimes compensate for damaged areas. New therapies that enhance plasticity, combined with targeted training, are improving outcomes for stroke and traumatic brain injury. Understanding the mechanisms of plasticity and how to optimize it therapeutically remains a major focus of neuroscience research. And here, as we near the end of our journey, let's pause. We've traveled through structures and systems, through memories and emotions, through consciousness and mystery. We've explored how neurons communicate, how memories form and fade, how sleep knits together the experiences of the day. We've seen how language emerges from distributed networks, how emotions color experience, how the social world shapes the brain from infancy onward. But perhaps the most remarkable fact of all is that your brain, this 3-pound universe, generated every thought you had while listening to these words. It created the experience of understanding, of curiosity, perhaps of wonder. It allowed you to contemplate itself, a self-referential loop that remains one of nature's most profound achievements. Your mind isn't separate from your brain, nor is it reducible to mere neural firing. It's the emergent property of billions of neurons working in concert, shaped by evolution, sculpted by experience, unique to you, yet fundamentally connected to every other human mind through our shared biology and common humanity. As you drift towards sleep, your brain will continue its work. It will consolidate the memories of today, clear away metabolic waste, replay experiences, and prepare for tomorrow. It will dream, generating entire worlds from pure neural activity. And throughout, it will maintain the essential rhythms that keep you alive, the unconscious processes that continue while awareness fades. The human mind remains, in many ways, the greatest mystery we can investigate. Each discovery opens new questions. Each answer reveals how much we have yet to learn. But this incompleteness, this vast territory of the unknown, is part of what makes neuroscience so compelling. We're studying ourselves, mind examining mind, consciousness investigating consciousness. You carry within you something more complex than anything else we've found in the universe, something capable of love and creativity, of reason and imagination, of understanding its own workings while remaining mysterious to itself. Your brain, your mind, your consciousness, whatever we call this remarkable phenomenon, is both you and what allows there to be a you at all. Rest now, knowing that your brain will continue its essential work while you sleep. Dream deeply, and when you wake, you'll carry forward this extraordinary gift of consciousness into another day of experience, learning, and wonder. If you're still awake, you can continue with another exploration. If not, I hope you sleep well and wake refreshed. perhaps with a deeper appreciation for the magnificent complexity that makes you who you are. Thank you for listening. Good night.